Yeah.
Good afternoon, everybody. My name is Andrew Muir, Science Director of the Great Lakes Fishery Commission. I'm sitting in for Dr. Bo Bonnell at USGS Great Lakes Fishery Commission, who normally leads these workshops or these webinars. Bo is currently out on holiday. So he is the coregonine Science Program Director. Welcome to everybody. This is the fourth webinar in the series here. We've had pretty good interest and strong participation across the board for these. I want to make a couple of quick announcements. Nick has put the recordings of the previous webinars, the presentations, up on the Great Lakes Fishery Commission YouTube channel. So those are available. I think he's going to paste, and he did there, paste the link to that channel down in the chat for those of you who are interested in some of the previous presentations. Also want to announce that the next webinar will be hosted on August 6th, and we've got Randy Brown from the U.S. Fish and Wildlife Service in Alaska talking about Coregonus lorette, the Bering cisco, spawning migration. So that'll be of interest, a species outside of the basin, obviously. There may be some interesting things we can learn there. So the talks we have today, two presentations around a similar theme of stocking. The first presenter will be Nick Boucher from the Great Lakes Fishery Commission. He's our Fishery Research Program Associate. He's going to be presenting a reanalysis of a data set from a project that was led by Chuck Bronte and Matt Cornice at U.S. Fish and Wildlife Service on global coregonine stocking efforts. That will be followed by a presentation by Ben Rook talking about how many ciscos could be required to stock the Great Lakes from a historical perspective of populations that used to support fisheries in the past. So with that, we'll start with Nick, and we'll take a few questions after Nick's presentation, and then we'll have a bit of a discussion after Ben's presentation. Okay, go ahead, Nick.
Thanks, Andrew. Yeah, so can everybody see my screen okay? And any feel free to interject if there are any issues. Yeah, so as Andrew said, I'll be taking you all through a reanalysis of some work that started before I began at the commission. And it was a retrospective analysis of coregonine stocking efforts throughout their global range. And hopefully this will inform future restoration efforts throughout the world. So just as some background, in the Laurentian Great Lakes, there is extensive stocking of lake whitefish as well as lake herring or Coregonus artidi that began in the late 19th century as early as 1870, and it continued through 1960. Tom Todd reports that there were over 32 billion individual stocks during that time period, which included about 6.5 billion Coregonus artidi. And so here is the earliest source that I was able to get my hands on. It's from Rygaard in 1910, and it is an examination of stocking throughout the Laurentian Great Lakes from the late 19th century. So here he reports that planting of lake whitefish juveniles was ongoing in all 5 lakes as early as 1892. And then he goes on to actually make an attempt at finding adequate stocking density, for lake whitefish. And he found that planting about 30,000 juveniles per square mile, or roughly 100 juveniles per pound caught, correlates with an increase in about 72% in the catch. So even in the late 1890s, the stocking program was relatively controversial, with opponents arguing that artificial propagation had no positive effect and was actually the cause of a negative impact through damage to spawning fish. And Tom Todd, in that source that I referred to earlier, actually revisited this analysis, and he went on to find that Rygaard's conclusions were actually incorrect. So by the 1960s, stocking of coregonines, both whitefish and cisco, was largely shelved due to lack of evidence that it was effective. Conversely, stocking of coregonines in both Scandinavia and Western Europe has been widespread since the the late 18th century. I found sources reporting that there was some small-scale stocking of glacial tarns going back as early as the 1790s. And the FAO from the UN reports that hatchery operations in Western Europe began as early as the mid-1800s, and they were developing guidelines throughout the 18th and 19th century. So contrary to U.S. stocking, stocking of coregonines has remained widespread throughout this period. For example, in Lake Constance in Germany, stocking began in 1887, and it continues today with an extensive fry stocking program. And examples like Lake Constance have led to advances in technology and practices that we could learn from here in the United States. So with that in mind, we came to the conclusion that there's a wealth of information on rearing and stocking coregonines abroad, as well as some examples from the Laurentian Great Lakes. But these data are not readily accessible or in a usable format for a meta-analysis like this. So our primary objective was to generate a robust data set that identifies key factors in determining the success of coregonine stocking throughout their global range. And here we use measures of success of survival where not previously detected, or reproduction of stocked individuals. And then we went on to use this data set to try and identify some key considerations for managers who are undertaking a coregonine stocking program or considering embarking on a stocking program like this. So the first step of this analysis was to conduct a literature search. The literature search was done through Google Scholar, Web of Science, and the U.S. Fish and Wildlife National Conservation Training Center archives, using keywords related to coregonines and stocking. Each source was evaluated for the binary response variable, survival or reproduction, as well as reviewed for independent response variables. So here's an example of a paper from 2004 that was reviewed. And there is within that paper, there was information about stocking in Lake Baldegg, which is in Switzerland. And it reported the stocking density as well as some other system-specific variables. So the result of this review was a data set encompassing 67 papers that were from as early as 1897 and as recent as 2015, and 371 stocking attempts or individual data points. So each source, as I mentioned, was evaluated for predictor variables that were then processed. First, variables with no clear patterns, or those that were recorded less than 50% of the time, were removed. And then for predictors that were highly correlated, here meaning a Pearson's R of greater than 0.65, we removed all but one of the predictors. So for example, we found that the predictors country, region, and species were all highly correlated. So we removed country and region while retaining species because we felt it was the most biologically relevant factor. For numerical values, we replaced missing data with the geometric mean. This is opposed to the regular mean because our data were not normally distributed. And for categorical data, we replaced missing data with an unknown category. So hopefully our predictor variables are relatively straightforward, but I'll provide some brief definitions for the ones that aren't self-explanatory. So stocking purpose, we had 3 categories: restoration, augmentation, or introduction. So restoration of a population, augmentation of an existing population, or introduction where coregonines were not previously present. For trophic status, we had oligotrophic, mesotrophic, or eutrophic. System type, we looked at lotic and lentic systems. For coregonine population status, or here population status, we categorize them into abundant, rare, or extirpated. And for fish thermal gild, that is the thermal gild of fish occupying the water body that stocking is occurring in. And then also life stage stocked, we identified embryos, larvae, age 0 juveniles, or year 1 juveniles. And we then fed these predictor variables into a boosted regression tree model for 2 different response variables. So as I mentioned before, survival and reproduction. So our data set encompasses 2 subgenera within the genus Coregonus, that Louis Bernachet identified in 1991. So first was Coregonus, or the true whitefish, which are characterized by a low gill raker count, subterminal mouth, and a benthic feeding habit, or the subgenera Leucichthys, which is characterized by a terminal or superterminal mouth, high gill raker count, and a pelagic-oriented feeding habit. So as Andrew mentioned in the introduction, this is actually a reanalysis of previous work. In the previous work, reviewers raised concern that it was inappropriate to combine the whitefishes and pelagic ciscos because they have different life histories. So ultimately what we decided to do was to split them into 2 data sets, with the end goal of being able to identify a suite of variables that were important across the genus Coregonus for consideration. So for this analysis, we used a boosted regression tree. Boosted regression trees are a combination of CART models, which generate predictions based on decision trees generated through binary splits, and each split seeks to minimize the variance on either side of the node. Boosting is an artificial intelligence algorithm that uses the loss function of each individual CART or decision tree to generate the following tree in a way that best minimizes loss. So here's a rough visualization of each of a boosted regression tree generates numerous decision trees using each previous tree to develop the next one. And we chose boosted regression trees because they're robust to a variety of non-normal data. As I mentioned before, a lot of our data were not normally distributed. They can also accommodate both categorical and numeric predictors, which was good for our data set. They can fit nonlinear relationships and automatically handle interaction effects. And another important factor was that they help to identify the most impactful predictor variables. So after splitting our data set into pelagic coregonines and whitefish, and then evaluating both of these data sets for 2 response variables, we ended up with 4 total model iterations. And the first step of this modeling analysis was to conduct some sensitivity testing to identify the parameters that were under which our models were optimal. So tree complexity here refers to the extent to which interaction terms are fitted by specifying the number of nodes in each tree. So a tree complexity of 1 would mean there are no interactions modeled. A tree complexity of 2 means there would be two-way interactions, and a tree complexity of 5 would mean multiple levels of interaction. Learning rate is the parameter that determines how much each tree contributes to the overall model. And bag fraction is the proportion of the data that are randomly sampled to build each tree. So a bag fraction of 0.5 indicates that for building each tree, half of the data are randomly selected. So across our 4 model iterations, we found different parameters for each model under which the model was optimal. And optimal here is defined as having the highest cross-validated d squared, which is just a measure of the proportion of variation in response to the variables explained by the predictor versus the proportion of variance explained by random chance. So let me apologize for the next few slides. The font is going to be small, and there's going to be a lot of information to take in. My main goal here is to just give you an idea of what the model output looks like, and then I'll go into the results in a bit more detail afterwards. So the model generates partial dependency plots, which also include the relative influence as a percentage for each predictor. So here, this is the lake whitefish survival model iteration, and it has each predictor variable ranked in order of their influence. So number of years stocked was the most influential at 30.7%, and coregonine population status was the least influential at 0.3%. You can also see that there's a line for continuous variables and hashes for categorical variables. So here we have the relative influence of all of our predictor variables across the 4 model iterations. And so originally our goal was to identify a suite of predictors that was impactful across all 4 iterations. And in fact, we did see that 5 emerged that were either highly or moderately impactful across our 4 iterations. So on the left here, you see a brief description of the variable as well as its relative influence as a percentage. So both number of years stocked and lake size were highly or moderately influential across all 4 model iterations. Stocking purpose, thermal gild, and species were either highly or moderately influential across 3 of the 4 model iterations. So given the influence of these 5 variables, we think that they likely represent important considerations for managers undergoing a coregonine stocking program. So here are the 5 parameters that I identified before in what I hope is a bit more of a digestible format. So in the leftmost column, we see a description of the variable, and in the columns on the right, there's their relative influence for each model iteration. So as I said before, it's not necessarily feasible in the scope of this talk to go through each partial dependence plot. So there were 10 for each model iteration, so we had 40 in total. So instead, what I'll do here is take you through how I interpret each one, and then I'll report the effects in a simpler format. So here we see a strong, so on the y-axis, we have the marginal effect of the predictor variable on the response, here survival. And on the x-axis, we see the predictor variable, which in this case is number of years stocked. So we see a strong negative impact when stocking for less than 5 years. Note the scale is it goes down to negative. There's a slight positive or neutral impact for 10 years or longer. So we think that this might be due to weather effects. We've seen that high wind years are followed by weaker year classes of coregonines, and weather conditions had a more dominant effect on year class strength, as Tom Todd described in 1986. So hypothesis explaining this effect might be that stocking for shorter periods of time make these programs more vulnerable to aberrant weather patterns. When looking at pelagic coregonine survival, we actually see the opposite effect for number of years stocked. So there's a strong positive impact when stocking for less than 10 years, with a slight negative impact thereafter. So one explanation for this might be that there's a compensatory growth of piscivores following stocking. So one example in Lake Ontario, following cisco introductions, both abundance and size of lake trout increased, which in turn led to greater predation on ciscos. Additionally, pelagic ciscos have smaller body size than whitefishes, so potentially they're more vulnerable to predation. So that might explain why we're seeing opposite effects for something that should be relatively similar. Additionally, we think that this might be skewed by stocking programs that were continued due to their popularity, despite poor evidence that they work. So the stocking programs that I talked about in the introduction between 1870 and 1960 were largely continued on the basis of positive testimonials from stakeholders and fishermen, despite dubious evidence that they actually accomplished any of their stocking goals. So here is a summary of the effects that we see for number of years stocked across all 4 model iterations. So I've already talked about whitefish survival and pelagic coregonine survival. But for whitefish reproduction, we see a similar effect where over longer time periods, whitefish reproduction was less likely to occur. And for pelagic coregonine reproduction, we see that there is more likely to be reproduction over longer time periods. For water body size, it had a negative impact on survival of both whitefish and pelagic coregonines, while having a positive impact on reproduction. So here what I'm saying is that in larger lakes, both whitefish and pelagic coregonines were less likely to survive, while in those same large lakes, they were more likely to reproduce successfully. So I reviewed some of the papers within our data set to figure out why this might be. And a lot of them commented that dredging and lake level regulation had a strong impact on spawning habitat, particularly in lakes that are 115,000 hectares or less. Additionally, the patchiness of large lakes may lead to more dispersed feeding opportunities and thus poorer survival. But the explanation for the improved reproduction might be that their larger volume allows them to modulate the effects of habitat modification, and additionally, patchiness may provide a broader diversity of spawning habitat. For stocking purpose, stocking for introduction seemed to be the most successful across all 4 model iterations, which was interesting to see. And we think this, and one source that I reviewed found that stocking with whitefish larvae was reported to fail if a naturally reproducing population already existed. And this is possibly because of intraspecific predation and competition. Additionally, we found that translocating Coregonus lavaretus, the European whitefish, into a new water body in Scotland resulted in a new population that had a lower parasitic load compared to the original water body. So this indicates to me that the lack of coregonine hosts in the receiving water body may have eliminated a bottleneck, in this case parasites, to the reproduction or survival of the introduced fishes. For fish gild of the receiving water body, cool and cool warm were most successful for whitefish survival. Cool warm and cool were the most successful for pelagic coregonine survival. Cool and cold cool thermal gilds were most successful for whitefish reproduction, and cold and cool warm were most successful for pelagic coregonine reproduction. So this is surprising because coregonines are classified as a cold water fish. We think here maybe the marginal habitat may be overcome by other factors that are aiding in their survival or reproduction. And lastly, species stocked. So across 3 of the 4 model iterations, we see that the European species are most successful. So in our whitefish data set, Coregonus lavaretus, which is the European whitefish, was most successful. And in pelagic coregonine survival, European species were most successful. We may attribute these differences in success to higher levels of rearing and stocking in Europe that I alluded to earlier. And this is actually a good sign. It shows that both experience and improved technology may help to overcome some other factors. So to wrap up with some conclusions, one of the more surprising findings that we had was that bigger does not equal better, or stated more clearly, we did not see across any of our model iterations that that life stage stocked had a strong impact. So the prevailing wisdom has been that older life stages are typically more likely to survive and reproduce following stocking, but that was not emergent from our models. So that's not to say that life stage is not important, but perhaps this indicates that other factors can help to overcome challenges with stocking earlier life stages. Secondly, higher levels of rearing and stocking experience may help to overcome other factors. We can see this when species seem to be confounded by geographic location, with European species having a higher likelihood of success. So experience in rearing and stocking may help to overcome either system or species-specific challenges. And lastly, I'll take you all through a case study of coregonus artidi restoration in Saginaw Bay. So just to summarize, gamete collection for this project began in 2016, and stocking by the Michigan DNR began in 2018. And they have a stated 10-year benchmark of evidence for natural reproduction and offshore pelagic existence. So since the ultimate goal of this project is to generate a reproducing population, for this, I'll look at the pelagic coregonine reproduction model iteration. So here we see that over a long period of time, there's a slight positive effect with increasing the number of years stocked beyond 10 years. So this is that's a good sign because the MDNR's stated plan includes goals for 10 years and beyond. So it's important to consider the time and money investment when embarking on a program like this. Additionally, there's a positive relationship between lake size and coregonine reproduction. So at very small lake sizes, around less than 100,000 hectares, we see a strong negative effect of lake size. So Saginaw Bay is roughly 296,000 hectares. So that's another positive. Lastly, we'll look at the thermal gild of Saginaw Bay. So on this partial dependency plot, the thermal gilds listed are in alphabetical order. So leftmost we have cold, then cold to cool, then cool, and then cool warm. You can see that cool warm was the second most successful after unknown, which we'll just ignore for the purpose of this analysis. And I would classify, I haven't done any quantitative analysis of the thermal gild of Saginaw Bay, but given that they have a strong walleye and perch fishery, I would classify it as a cool warm fish gild. So in hindsight, our analysis indicates that the decision-making surrounding cisco restoration in Saginaw Bay is supported by our model findings, and there's a likelihood that this program will succeed. So with that, I'd like to thank you all for your attention, and I guess we have a few minutes for questions.
Yes, thanks, Nick. Nicely done. I see Chuck Bronte has a question. Go ahead, Chuck.
Yeah, nice job, Nick. Just wanted to clarify that the program in Saginaw Bay, that's not just Michigan DNR, but that's a program that was approved by the Lake Huron Committee, which involves the tribes, the state, and the Ontario Ministry. And those fish are actually reared at one of our U.S. Fish and Wildlife Service hatcheries, but it is a collaborative program.
Yeah, thanks for clarifying, Chuck.
Great. Any other questions? Taylor, go ahead.
Hi. I know you said that you had to drop country and region because they were correlated with species, but I'm wondering if you thought about fitting models with either country or region instead and compare those to the species specific models to see if any of the variable importance results vary. I'm thinking about like anything that might come out that could potentially reflect different management strategies by different agencies that resulted in any sort of success for those working on the same species.
Yeah, that's an interesting thought. I don't think that we have considered that, but certainly sounds like something that's doable. Like I said, so I started on a reanalysis of this. So maybe Chuck or it looked like Matt was on the call as well. So maybe they can correct me if I'm wrong, but I don't think that's something that we've considered yet.
So Nick, I would just add that my memory is a little foggy, but I think early on we did have a model that had I know country in it, and basically we saw higher success from the European nations compared to North America. But the species were so highly correlated there. You know, I think in an early edition of the draft we were talking about that correlation and how the, you know, is it a species effect? Is it a management practice effect? But it's really impossible to disentangle, and I think, at least my own opinion, is that biologically species seems to make a little more sense, but that's up for debate, I suppose.
Okay, great, thanks.
I just wanted to add to that, if I may. So just so the audience understands, maybe not everybody does, but you know, even though these coregonid programs ended largely in the last century in North America, there are only really two, actually now three, actually one long-term whitefish augmentation hatchery in Ontario that serves Lake Simcoe. So they have probably the most long-term experience, and that's been largely successful. And then the Leech Lake Band in Minnesota has been stocking lake whitefish as well. But clearly the Europeans have way more effort and expertise in this, so much so that we, after the coregonine symposium in Austria, we put together a team actually under the same grant from the commission to go over to Finland and actually do a stem to stern survey of their coregonine culture practices.
Okay, great. One last question and then we'll move on. Roger Knight posted in the chat, how is survival defined and reproduction for the two different groups? And we did have very specific definitions of those. I don't know if you have them on the top of your tongue or if you want to post those in the chat in response to Roger's question.
Yeah, maybe I'll post them in the chat just because I want to revisit the paper and make sure I get it correct. I think for survival, it was detection of individuals where they weren't previously detected. And for reproduction, I can't quite remember. I think, especially with the older sources, there, you know, there wasn't always marking of stocked individuals or anything. So I think we had to get a little bit creative with how we evaluated that. But I'll pull it up and post it in the chat.
So just if I can add, the impetus for this work was, you know, here we're embarking on a large scale stocking program of a species that, you know, we got plenty of experience with salmonines in the Great Lakes. I mean, that and perkids, but not with coregonines anyway recently. So the idea was to look to see what worked in Europe, because I think there was some buzz about the thought that the probability of this working in the Great Lakes with coregonines was really low. And that was probably, those judgment calls were probably made without knowing what was going on in Europe. And certainly our attendance at the coregonid symposiums going back 10, 15 years or longer suggests that we need to look across the pond. That's all I'll say.
Okay, thanks, Chuck. All right, any other questions, just post them in the chat and we'll get to them. Let's move on now to Ben.
Can everybody hear me okay?
Yep.
All right, so Nick talked a little bit about some of the factors that determine success or failure. I'm going to go ahead and try and put some numbers on the Cisco restoration program throughout the Laurentian Great Lakes, primarily in Lakes Huron, Erie, Ontario, and then Michigan. So basically everything outside of Lake Superior. I'm going to get into some really big numbers later on in the presentation, but just keep in mind that these are fairly realistic when you go through and compare these to what we see for stocking densities for some of the European species. So prior to European settlement, cisco and deepwater ciscos were the most abundant and ecologically important fish species in the Great Lakes, but nearly all populations were reduced or extirpated basin-wide by the 1970s due to overexploitation, eutrophication, and then interactions with non-native species. So basically what we see are these commercial harvests that are declining throughout the basin as these species are going and being replaced by non-native alewife and rainbow smelt. So the figure I have provided here is just an example. It's for cisco and deepwater cisco harvest combined for all of the Great Lakes. In any particular lake, this could happen up to 50 years earlier, a few years later, but it's just a good general example of what went on in each of these individual lakes. So when I talk about cisco in the rest of the presentation, I'm going to be talking about our T-dye. And then when I talk about deepwater ciscos, I'm basically going to be talking about all the chub forms, so bloater kai-eye, blackfin cisco, and shortjaw cisco. And I'm going to kind of take a general approach in the background material and then kind of narrow it down to cisco later on. So after we saw these declines for all these coregonous complex forms, we see that bloater persisted in Lakes Huron and Michigan, but most of these other deepwater ciscos were either extinct, extirpated, or hybridized in pretty much every other lake besides Lake Superior. Currently, we have bloater and kai-eye along with shortjaw cisco in Lake Superior, whereas blackfin cisco are kind of an uncertain status. We also have remnant cisco populations that persisted in Lake Superior, Huron, Michigan, and Ontario. But with the exception of the populations in Lake Superior, most of these are really pretty small and isolated compared to what they were historically. In Lake Superior, cisco partially recovered after the 1980s, but contemporary abundance is likely below historical levels, and it's really driven by these sporadic recruitment events. So I just want to mention these forms that you see here listed because these are the ones that are considered extant by Eschenroeder et al. 2016. I believe one of these, blackfin cisco, is probably Lake Nipigon. But these are the forms that we could be targeting for restoration at some point in the future moving forward. So as a complex, these were all really important because they were locally adapted. They served as high quality forage for native lake trout. And at least compared to what they were replaced with, they supported fairly extensive high value commercial fisheries. Now, if we look at what they were replaced with, primarily alewife and rainbow smelt, they're prone to die-offs. They're really low quality forage. They contain thiaminase and cause EMS. And they only support really limited low value commercial fisheries, kind of thinking of the smelt fishery in Lake Erie right now as an example of that. Since the mid-2000s, though, we've seen some ecological and environmental conditions that have changed throughout the Great Lakes, and these are thought to have improved conditions or at least made them more favorable to coregonids compared to alewife and rainbow smelt. So what we've seen specifically are declines in alewife and rainbow smelt populations compared to what they were prior to 2000. This could lessen natural mortality through reduced competition and then predation, primarily during the larval stage. Similarly, tongue-tied today. We've also seen commercial fishery regulations that have either reduced or eliminated fishing mortality for many of these populations that were heavily exploited prior to the 1970s. We've also seen some oligotrophication in Lakes Michigan, Huron, and Ontario, to where we have similar levels of primary production to what we see in Lake Superior, which currently supports the largest cisco populations and also the most diverse coregonid assemblage in the Great Lakes. And another really important factor here are environmental conditions for overwintering eggs, primarily bottom dissolved oxygen concentrations, which have also improved in some of these historically important nearshore spawning and rearing areas like Green Bay and Lake Michigan, and then likely Saginaw Bay and Lake Huron as well. So some of these areas that were really highly degraded in the mid-1900s may be suitable for reintroducing and restoring some of these populations that used them in the past, and kind of specifically thinking about cisco here. So stocking is expected to play a major role in reestablishing self-sustaining cisco and deep water cisco populations in most of the Great Lakes. But we do have these questions regarding hatchery propagation that are research priorities before we can kind of go ahead and ramp up production even more than what it already is. So I have cisco and then bloater put up here. These are the two forms currently being stocked, cisco in Lakes Huron and Michigan, and then bloater in Lake Ontario. Nick kind of mentioned this a little bit earlier, but stocking success generally increases as a function of life stage or size stocked for older, larger fish and coregonids. But these older, larger fish do require more care, infrastructure, and then time in hatchery. So this is kind of, more money and more manpower as an investment. So as managers, we kind of need to carefully weigh the potential costs and benefits of rearing fish to these more advanced life stages. So for example, the above two species are generally stocked as 10 to 11 month old fingerlings. So there's, some substantially greater rearing costs associated with those when you compare those to fry. And one of the things that's really missing are densities or numbers of fish required for stocking to guide these restoration efforts. So for most of these populations, we really don't have any, target goals for what we need to see to guide these restoration efforts. We're just kind of putting fish in and hoping they do okay. So this brings us to our objectives. So for this particular study, we wanted to determine the number of ciscos needed for stocking at 3 different life stages and then a range of spatial scales to mimic what we see for historical and contemporary age 1 and adult recruitment rates in Lake Superior. So when I talk about Lake Superior, I'm specifically talking about Wisconsin waters from here on out. And this is one of the more healthy populations in the Great Lakes, so it's kind of a good reference point to use. So for our 3 life stages being stocked, we want to look at fry, fall fingerlings, and then the age 1 life stages. And then for our spatial scales, we want to look at the basin-wide scale, except for Lake Superior, the lake-wide scales, and then individual stock scales. So one of the other things that's been pointed out in workshops before is sort of this need to identify some of the historically important cisco spawning and rearing areas or stocks to prioritize where we do put these fish when we start ramping these programs up. Because we do have limited coregonid production capacity in hatcheries. So I'm also going to talk a little bit more about some of the more important areas historically later on as well. And then, this is a Cisco specific project, but the idea here was to provide a generalized framework that with a few changes here and there could potentially be described or could potentially be used to describe future stocking needs for some of the deep water Cisco species like bloater and kai maybe. So our general approach was to use Ricker stock recruit parameters Rmax and Smax, which were previously estimated for historical and contemporary ciscos in Wisconsin waters at Lake Superior as our age 1 and adult restoration targets. So if you can kind of picture the Ricker curve, it rises up from the origin, it reaches a peak, and then it declines. So these are the parameters estimated from the peak of that. So kind of good age 1 and adult restoration targets. Our Rmax is basically our peak recruitment or age 1 numbers. And then our Smax is going to be our spawning size that produces that, or our adult numbers. So we wanted to kind of take a bookend approach to this. So we looked at both historical and contemporary parameters. The historical parameters are from the pre-55 period. So you've got more phosphorus in the system and more ciscos. And then the contemporary period is 1992 to 2016. So lower productivity, less phosphorus in the system, and likely less ciscos as well too. I do want to mention that based on changes in productivity more recently in Lakes Huron, Michigan, and Ontario, They're very similar to what we see in Lake Superior right now. So some of these contemporary parameters are probably more likely applicable to those lakes, whereas some of these historical parameters are likely more applicable to Lakes Erie and St. Clair, given higher productivity. And I do include Lake St. Clair here just because there were some migratory spawning populations that use that historically for cisco. So the idea here is we're going to use our Rmax and our Smax as targets. We're going to scale this to suitable age 1 cisco rearing habitat area and get fish per hectares less than or equal to 80 meters depth. That's generally their preferred spawning and rearing habitat in Lake Superior. And it's important to point out here that this assumes there's no other limiting factors in any of these other lakes, such as unsuitable oxythermal conditions. So really just a broad description of rearing habitat area. And, you know, this may not be true in all cases in all areas of the lower Great Lakes. Kind of thinking of Eastern Lake Erie here, where oxythermal conditions may be a little less than ideal. So kind of continuing with this general approach, we're going to estimate lake-specific instantaneous natural mortality rates, or M, using the Pauli equation. So this basically incorporates lake-specific air temperatures and then von Bertalanffy growth parameters, which we're using previously estimated parameters for both historical and contemporary ciscos in Wisconsin waters of Lake Superior. And then we're using a randomized version of this equation and bootstrap methods to back calculate lake-specific stocking densities for each life stage. So fry, fall fingerling, and age 1 from our age 1 and adult restoration targets. So Rmax and Smax. I mentioned we're using bootstrap methods, so we do get 95% confidence limits on these. I'm just going to be presenting median values today, just so we don't get too cluttered with some of the figures. And then once we have these estimates, these are going to be in density. We're going to go ahead and apply these lake-specific densities to areas identified for individual stocks using USGS agency records and then Goodyear et al. 1982. So our agency records are based largely on Goodyear et al. 1982, Kelps 1929, and then Michigan DNR agency records as well. And then Goodyear 1982 is just the spawning atlas for the Great Lakes. So I'll go through an example here just for our age 1 targets. So basically what we're doing is we're taking our Rmax, we're scaling that to suitable cisco rearing habitat area in Wisconsin waters of Lake Superior. We're estimating a lake-specific random mortality estimate for each of the lower Great Lakes. And then we're applying a certain number of iterations of natural mortality to establish fry, fall fingerling, and then age 1 stocking densities. So for our age 1 fish, no natural mortality is applied. We're just assuming we're stocking those, and we're going to mimic what we saw for densities in Lake Superior. Our fall fingerlings get 1 iteration of natural mortality, and then our fry get 2 iterations of natural mortality. I do need to mention we're probably substantially underestimating natural mortality experienced by fry. If you go back into the literature, you know, their survival is substantially less than what you see for fingerlings and then age 1 fish. And I'll talk a little bit about this later on. So once we have our densities, we're basically scaling to stock specific areas in the lower Great Lakes, areas less than or equal to 80 meter depth. And then this gives us a stock specific number of fry, fall fingerlings, and then age 1 fish. So once we get these, we basically combine these at the individual lake scale, or we combine these at the basin-wide scale to get our total numbers for stocking. Same general approach for adult targets, but we're using weighted average adult ages of 4.2 years and 5.5 years for historical and contemporary ciscos. So when this is all said and done, we get 12 different back calculations or 12 different stocking scenarios for each area or individual stock. So to kind of break this down a little bit, we have 2 targets, our age 1 and our adult targets, our Rmax and Smax. We've got 2 periods, historical and contemporary. And then we've got 3 life stages for each of those. So fry, fall fingerling, and then age 1. So study area, just in general, is pretty self-explanatory, entire Great Lakes Basin. If we take a look at the points, these are the locations of the land-based air temperature monitoring stations used. For the east-west running lakes, we tried to pick, 3 or 4 points running across the entire length. of each lake. And then for the north-south lakes, we tried to pick some in the north and then some in the south, just so you get a good average temperature. And the unshaded white areas here are the 80-meter depth contours. So you can think of all of the white areas in this particular map as being potential spawning and rearing habitat area for cisco. And again, this is a pretty broad description. So one of the other things we needed to do was to establish areas for individual stocks for spawning and rearing. So this particular map shows those. If you take a look up by the MN abbreviation, we also see WISC. So that's Wisconsin Waters of Lake Superior. So we're taking data from that area and then kind of applying it to the remainder of the lower Great Lakes. And these were identified again using USGS agency records and then Goodyear et al. 1982. So we have general spawning locations and rearing locations. And then we're going to go ahead and put a 25-kilometer buffer on those. And then we're going to use that to estimate our suitable rearing habitat area. So these are pretty rough estimates, but it's a good generalized approach that allows us to combine data from all these different sources. And again, I mentioned basically estimating numbers for each of these individual stocks and then scaling them up for lake-wide and then basin-wide estimates. So kind of to get into some results, differences in lakes were really driven by differences in air temperature, which thus drives instantaneous natural mortality estimates. So the warmer lakes generally have higher estimates of natural mortality and require higher stocking densities, whereas the colder lakes require lower stocking densities. So I have these listed from warmer to colder, from left to right. And then I've got our adult equivalents on the top, and then our age 1 equivalents on the bottom. So these are the stocking densities that would be required in each lake to mimic what we see in Lake Superior based on differences in natural mortality. So just kind of in short, stocking scenarios that mimicked contemporary age 1 recruitment rates. So again, lower productivity time period required the lowest densities, whereas our stocking scenarios that mimicked historical adult recruitment rates required the highest stocking densities. So if you kind of think about the differences there, we have higher productivity historically, and then we're also applying more iterations of natural mortality from the adult stage back to what we stocked versus the age 1 stage. Just in general, stocking densities based on historical recruitment rates were about 9.4-fold lower than those based on contemporary recruitment rates. And then stocking densities based on adult recruitment rates were about 1.6-fold greater than those based on age 1 recruitment rates. And I thought it would be useful to compare these to some of the published densities for European coregonus species. So if we take a look at those up in the corner, we see on average they're about 2,500 fry per hectare, 350 fall fingerlings per hectare, and 25 age 1 fish per hectare. So these estimates generally bookend those, except for the fry life stage. And again, we're probably substantially underestimating natural mortality just because of the way we applied that. Our estimates for the fingerling life stage and then the age 1 life stage are probably more accurate. I think our contemporary estimates are probably a little bit on the low side when you compare them to the European species, and then a little bit on the high side for the historical estimates. And again, these are values of 1,000 bootstrap samples. So these are the medians. So I thought the most informative thing here would be to put out some basin-wide numbers. So where we're basically going in and combining the total number of fish needed for each of these individual stocks at the basin-wide scale. I'm going to focus on minimum values for fall fingerlings, since this is kind of the adopted preferred life stage by most agencies. And again, these minimum values correspond to stocking scenarios that mimic contemporary age 1 densities in Wisconsin waters of Lake Superior. So this is pointed out by the red arrow you see here. So if you want to take and simultaneously approach this from a basin-wide scale, it would require about 500 million fall fingerlings. So just to put this into perspective, the estimated long-term maximum coregonid production capacity for all hatcheries throughout the basin is only about 4.5 million fish. So if you take 4.5 million and divide it by 500 million, you get this very, very small percentage you see here. So from a management standpoint, simultaneous stocking throughout the basin really isn't feasible under current or planned coregonine production capacities. So I think from a management standpoint, it makes much more sense to concentrate on one individual stock at a time. So some of the more important ones that are identified in the literature and then also contributed pretty substantially to historical commercial fishery production are some of these starred areas you see here. So the red stars are areas that were historically important, but are not currently being stocked. And then the yellow stars are, areas that were historically important that are currently being stocked. So if you go into the literature, you kind of have these accounts, and then you can also look at numbers harvested back before declines. And you see that Saginaw Bay and then Thunder Bays and Lake Huron were important. Green Bay, incredibly important to Lake Michigan. The islands near Sandusky, Ohio, and Western Lake Erie, and then near Hamilton, Ontario, and Bay of Quinte in Lake Ontario. And we also have additional stocking that's already occurring in Grand Traverse and Little Traverse Bays in Lake Michigan as well. So I kind of wanted to put the current Saginaw Bay program into some perspective as far as stocking estimates go. If you go by these estimates and just look at the minimum number of fall fingerlings required based on the minimum stocking scenario. So again, contemporary age 1 targets for Wisconsin waters of Lake Superior. Saginaw Bay would require a minimum of about 7-fold more fall fingerlings than we can actually produce given current plans. So moving forward, I don't want this to be like a doom and gloom presentation. We have seen some pretty substantial reductions in Pacific salmon and lake trout stocking rates, which have resulted in potential excess rearing capacity. I mean, it's probably open to debate whether these can actually be used to produce coregonines or whether we have the money to run those. But just to kind of throw some numbers out there, if you take a look at what's been stocked in Lake Huron in the past and compare it to the present, we've reduced that by about 5.5 million fish. And then we've seen similar declines totaling about 15.8 million fish in Lakes Michigan, Erie, and Ontario. So there's some potential excess rearing capacity there that could offset some of the cisco production needs at the individual stock scales. But I think when you scale this up to a lake-wide scale or a basin-wide scale, we're probably not going to get there no matter what we do. And I just want to point out that in Lake Superior, cisco do produce these extremely large year classes from relatively small adult spawning stocks. So it may just be a situation where we need to concentrate available resources on, 1 or 2 individual stocks and stock for as long as possible just to keep adults in the system and then give them a chance to take advantage of, ideal environmental conditions for recruitment until they get the opportunity to kind of, restore themselves and then potentially move to other areas as well. So with that, I have some acknowledgments here. This was provided, funding was provided on the DOI coregonine restoration template. USGS agency data was provided by Hannah Schaefer. Mike Slattery did the GIS and mapping work. And then all of the co-authors from the previous paper made that study possible by providing assessment and commercial fishery data for Lake Superior. So with that, I will take any questions.
Okay, excellent. Thanks, Ben. If you want to stop sharing your screen, we are exactly at 1:30, but we started a couple minutes late while we were waiting for people to trickle in. So we have some time for questions. I see Chuck Bronte has his hand raised.
I will stop sharing as soon as I get it to show up. There we go.
Yeah,
Okay.
yeah, sorry. Yeah, just a clarifying point. So it's not like we didn't know the answer with regard to rearing capacity and need, but this was a request. We had a workshop, a DOI workshop with the commission. Geez, when was that? Maybe 6-7 years ago. And at the end of that workshop, this was an ask directly to Mike Hansen to come up with these estimates, kind of just to set the stage for to get something out there so people understood the relationship between need and hatchery capacity. So, but certainly the step-by-step approach where you're going to the most important areas that were important historically and doing this in a sequential fashion it's kind of, you don't need a model for that, but we built one anyway, so. Nice
Great.
job, man, by the way.
Thanks, Chuck. Any other questions or comments for Ben?
I didn't get away that easy.
I'm impressed you answered all the questions. All right, well, thanks everyone. A special thanks to our presenters. A virtual round of applause there for you. We appreciate your time and efforts. And thanks to everyone for participating. We will hopefully see you in a couple of weeks. Again, just want to mention the next webinar is August 6th, and we'll have Randy Brown from U.S. Fish and Wildlife Service Alaska talking about Coregonus lorette. So Nick did put the registration link in the chat. Please reach out to him if you didn't receive it or if you need further information. Thanks again. Nick, did I miss any announcements or anything, or